Hello dear students, welcome back to Chinmoye's YouTube channel. So we are going to start today a new lesson on bacteria and fungi. So I'll be teaching you as per your curriculum. What is there in curriculum? Bacteria and fungi. The economic importance is your lesson name. So there we are going to study in details about both the bacteria and fungi. So let us start today's episode. So at the end of this lesson, what we will be learning? We will learn what is bacteria. We should know something regarding its shape, size, structure, movement, its nutrition and respiration and reproduction, right? So we should know something about its size, shape, structure, movement, nutrition, respiration and reproduction. So today's episode we will be dealing with all these things, and in tomorrow's episode we will be studying about the economic importance of bacteria and harmful effects of bacteria. So if you want to know more about bacteria and fungi, so you need to subscribe to my channel so that you get the updates every day which I am posting as per your curriculum, as per your proceedings in your lessons, how you are proceeding. So to get the updates daily, please subscribe to my channel, share it with your friends, and don't forget to write in my comment box whether you are liking my channel or not, and what all inclusion I can do in my lessons, so that it will be more easier for you to learn my lessons. Okay, so let us start with today's episode of bacteria. So, what is bacteria? First, let us discuss about what is bacteria. So, bacteria is the most simplest and the primitive unicellular organism without a true nucleus. Okay, so they are called prokaryotes. They are the most simplest and primitive unicellular organism without a true nucleus. Now, they belong to kingdom Monera. So you have already studied that classification part where I have dealt with kingdom Monera, Protista and all. So if you have not yet gone through that lesson, please go through it first. I'll put the link in the description box as well as in the I link above. Go through it. So then it will be easier for you to understand this bacteria. So this is under the kingdom Monera. So it is found everywhere: air, water, soil, food inside as well as outside our body. They lack nucleus and other cell organelles of complex cells. So they are lacking the nucleus and they are lacking other cell organelles of complex cells. They possess cell wall like the plant. Okay. So they are possessing nuclear material in the form of single chromosome, and which is not enclosed in a nuclear membrane. So the single chromosome which is inside the cell is not enclosed. It is open nuclear. It is a single chromosome. It is an open nuclear material and it is not enclosed by a nuclear membrane. So when I show you the structure of bacteria, you'll understand that how this single chromosome is not enclosed with a nuclear membrane. Now we are talking about the size. So the size, they are the smallest living organisms on earth. We all know that, and they are microscopic, so they can't be seen under the naked eyes. So what is their size? Their size varies from 0.5 to 5.0 micrometers. So their size is varying from 0.5 to 5.0 micrometers. So what is the calculation here? 1 micrometer, we can say that 1 micrometer is equal to 1 by 1000 of a millimeter. Okay, so 1 micrometer is 1 by 1000 of a millimeter, and because of this minute size they are not at all visible with our naked eye, so they are always microscopic. So bacteria is always microscopic. Okay, so it is microscopic, and 1 micrometer is equal to 1 by 1000 of a millimeter. So let us next come to its shape. Now shape, first we can say that according to shape of the bacteria there are 4 types or 4 shapes. So first we will take an example of the coccus. Okay, so coccus means it is we can say that coccus, it is cocci, it is the plural is cocci, it is plural is cocci. This means it is spherical shaped bacteria. Okay, this is coccus is a spherical shaped bacteria. Okay, now we can say that they are looking like bunch of grapes, and they are present in clusters like they are bunch of grapes. They are clustered, and they may also join end to end to form a chain. So they can also form chain formation. They can also stay in bunch or like grapes, and some cocci they also exist in pairs. So it differs from one group to another. Either they will be remaining in clusters like grapes, or they'll form end to end chain, or they can also exist in pairs. Now here is one clustered cocci. So this is an example of staphylococci or streptococci. You can say that they all staphylococci, they are in staphylococci. They are all present in a cluster or bunch like grapes. Now when we come to streptococci will remain in, if I take an example, streptococci, we can say that streptococci, they form a long chain. So they join end to end and they form a long chain. So they are joining end to end and forming a long chain. Okay, so that is streptococci. This is staphylococci. And another example is that diplococci, that exists in pairs. So another example is diplococci. So diplococci, they remain in pairs. Okay, diplococci, they are paired. So they remain in pairs. So staphylococci, it is like bunch of grapes. Streptococci, they are end to end, so they form a long chain. And diplococci, they are in pairs, but all are spherical shaped. So their coccus means spherical shape. Now next comes bacillus or rod shaped. So rod shaped bacillus, let us see how they are looking like. So they may be present as a single rod, or they are like E. coli or Escherichia coli, or in chain like Bacillus anthracis. Okay. So this is Bacillus anthracis. They are remaining in chain. So this is Bacillus anthracis. It remains in chain. Okay. And the next one is Escherichia coli. So this is E. coli. This is E. coli or Escherichia coli. This is single rod. Okay. They are forming single rod like structure. But the shape is rod. Here also the shape is rod. Here also the shape is rod only. They are remaining in chain forms, bacillus, and they are a single rod like structure. So this is the difference between two. Now next comes spirillum, or the plural is spirilla. So these are always spiral shaped or curved. Okay, they are spiral shaped or curved. So they have more than one flagella or one flagella at the end of their body. So example is Leptospira or spirillum. Now this is the spirillum. So these are all present in the bloodstream. So this spirillum is present in the bloodstream. This is an example of spirillum bacteria which is present in the bloodstream. Now next comes the vibrio or the comma shaped bacteria. So what is here? The body is curved only with one flagellum. So what is the criteria? The body is always curved with one flagellum. And example we can take the Vibrio cholerae. So how does it look like? It looks like this. So body is always curved with a flagellum, and this is an example of Vibrio cholerae. Okay, it is curved with one flagellum only. So now we will come to the structure of these bacteria. Now when we are discussing about the structure of a bacterial cell, we should know how it looks like. So this is how the bacterial cell looks like. So first we will see that now we all know that bacteria, they all lack chlorophyll, both bacteria and fungi, and their bodies are not at all differentiated into root, stem, leaves or flowers. And bacteria are single cell body, and whereas the fungi, it is a multicellular organism. Okay, so let us see that what is its structure like. So it is a very simple cell, and which consists of protoplast, that is the living substance inside is the protoplast, and it is surrounded by a non-living cell wall which is made up of peptidoglycan. So just remember this term peptidoglycan, it is not made up of cellulose. So as we know that in plant cell the cell wall is made up of cellulose, but in bacteria it is made up of peptidoglycan. Now beneath the cell wall we can see this is the cell wall, the outer covering. Beneath the cell wall is the plasma membrane. If you see the diagram here where I am pointing, where it is the plasma membrane or the thin cell membrane which surrounds the cytoplasm. So it is surrounding the cytoplasm. So the chromatin material is in the form of single molecule of circular DNA. So chromatin material, what is the chromatin material here? It is the nucleoid which is attached to the cell membrane. So this is the chromatin material present in bacteria, which is not at all, it is a circular form of DNA and it is attached to the cell membrane. Now in bacteria, Golgi complex and plastids are not at all present because they are absent, as it is a very simple type of cell. So no Golgi complex, no plastids are there. Now the chromatin material is not enclosed in the nuclear membrane, so it is free. So it is not enclosed in any nuclear membrane. Now some bacterial cells, they have capsules. You can see some bacterial cells are having capsules. So what does this capsule do? Which forms the envelope around the cell. So this capsule is actually helping the bacteria to remain in the dormant state. So meaning what is the dormant state? When the bacteria is not at all reacting to anything. So it is in a dormant state. It's not active. Now smaller rings of DNA known as plasmids. Okay, smaller rings of DNA known as plasmids. You can see here smaller rings of DNA called plasmids and ribosome granules. You can see here small ribosome granules which are also present in the cytoplasm. So cytoplasm contains small rings of DNA and ribosome granules also. So this is the structure of a bacteria, and you should practice this label diagram of bacteria with all the main important things which I have pointed out or told you. So the plasma membrane, the cell wall, the capsule, the flagellum, the plasmid, the nucleoid, ribosome, cytoplasm, fimbria. So these all should be there in this structure. So now we will move on to the next part. Now we will be dealing with the movement of the bacteria. So how does it move? So bacteria, they are immotile actually. They cannot move on their own. So very few bacteria such as Bacilli and Spirilla, those who are having flagellum whip-like structure at one end or throughout their body, they can move. Okay, they can move from one place to another. If they are not having this whip-like structure or flagellum, they can't move from one place to another. Now bacteria like Salmonella, they have flagella all over the body, and which helps them to move around in water. So Salmonella, that is having flagella throughout its body, and it is able to move from one place to another. Now we come to nutrition of these bacteria cells. So we know that they are not having any chlorophyll, so they cannot make their own food, so they cannot be autotrophic in nature. So they should depend on others for food, so they are heterotrophic. So they do not have chlorophyll, so their heterotrophic mode of nutrition is main. Now some bacteria, they draw nutrition from the dead as well as the decaying organic matters also. So they are drawing the nutrition from the dead and decaying organic matter, and hence they are saprophytic in nature. So they are drawing nutrition from the dead and decaying matters, so they are saprophytic. And some bacteria, they draw nutrition from the body of other organisms, that is they are parasitic. So they are making other organisms like host, and they are drawing the nutrition from them. So that is the parasitic nature of this bacteria. Now here comes respiration. So respiration in bacteria can be aerobic by absorbing oxygen present in the atmosphere, as well as anaerobic. So in absence of atmospheric oxygen, also some bacteria, they can survive. So that is anaerobic mode of respiration. So when we are talking about the reproduction in bacteria, we should know that how they are reproducing generally. So most of the bacteria, they reproduce generally by the means of asexual reproduction. So asexual reproduction, when we are talking about asexual reproduction, we should know the asexual method of reproduction is called binary fission. So most of the bacteria, they reproduce asexually, and a very few reproduce sexually. So asexual reproduction is by the mode of binary fission. So that is the endospore formation. So how it is happening, let us discuss this one. Now this one is a parent cell or a bacterial cell. So during fission, what is the thing happening? So bacteria, they divide into two. So cell division is preceded by the duplication of this genetic material, duplication of DNA. The bacteria grows, it is enlarging itself, the nucleus is enlarging itself, and due to DNA duplication, what is happening? The two DNA, they are pulled apart, they are being pulled apart. Okay, so the DNA material and the nucleus enlarges, and due to DNA duplication, two DNA, they are pulled apart into two sides, and two daughter cells are formed. Okay, so this wall grows, there is a wall which is growing and which is separating the two bacterial cells. So this wall grows, separating a single bacterium into two daughter cells, and in ideal conditions we can say that division is very fast. So this division is very fast. So very fast division. So how much fast? So some bacteria, they take only 30 minutes to divide. So they take only 30 minutes to divide into 2. So they take 30 minutes to divide into 2. Okay, so is it clear? So now when it comes to reproduction, we can say that it is very fast, and it is by the method of binary fission reproduction takes place, and it is mainly the asexual mode of reproduction in bacteria, and which is taking place in favorable conditions or by endospore formation. Is it clear? So just follow this pictorial representation which I have discussed here. So the parent cell, here the cell replicates its DNA. So the cytoplasmic region, this one elongates, and this replicated DNA, what is happening? The cytoplasm divides and cross walls form. So cross walls are forming here and two daughter cells are formed. So ideally it takes mostly 30 minutes to divide into two cells. So now comes the endospore formation on bacterial cells. So what is endospore formation? Endospore formation, it's not a method of reproduction in bacteria. So it means it is protecting themselves under favorable conditions. So under unfavorable conditions, we can say that certain bacteria which are gram positive, such as Bacillus, Clostridium, they produce spores within the cell wall. Okay, so these are known as endospores. So during favorable conditions, gram-positive bacteria, they are producing spores inside the cell wall, and these are known as endospores. So this is not a method of reproduction at all, and it means they are protecting themselves under favorable conditions. And endospores, these are all very much having great resistance to low temperature, high temperatures, pH changes, desiccation and effect of chemicals. So endospores, they are resistant, and when favorable conditions are they are actually reoccurring, these spores, they rupture and the dormant protoplasm, they become active. So when these spores are rupturing, the dormant protoplasm, they become active and the bacteria, they start multiplying. Okay, so under favorable conditions, we can say that they are reoccurring, the spore ruptures and the bacteria, they start multiplying. So let us see what is happening here. First, let us see, this is the bacterial cell. So these are the bacterial chromosome. Now the spore septum, they begins to isolate into newly replicated DNA and a small portion of cytoplasm. Now the plasma membrane starts to surround the DNA. So the plasma membrane is starting to surround the DNA. Spore septum surrounds the isolated portion. So two membranes are formed. Spore septum is surrounding the cytoplasm part. Then peptidoglycan, that is the cell wall layer, it forms between the membranes. So the cell wall layer is formed between the membranes here. Now here again the spore coat is formed and the endospore is freed from the cell. After the peptidoglycan layer is formed, spore coat is formed, it is freed from the cell. So an endospore example we have given here, the Bacillus anthracis, this endospore, this example has been given here. Now Clostridium also, they produce spores within the cell wall, and these spores are only called endospores. So there I have taken two examples, Bacillus and Clostridium, and here I have shown you the example of Bacillus anthracis. Now we should see that when there is a sexual reproduction, how these bacteria undergo sexual reproduction. Now sexual reproduction in bacteria, so how they are undergoing sexual reproduction, let us see. Now sexual reproduction in bacteria, it occurs through conjugation. Okay, so this sexual reproduction in bacteria occurs through conjugation method. So unusual form of reproduction in bacteria is the sexual reproduction. So what happens during sexual reproduction? During conjugation, the bridges are formed. You can see two bacterial cells are here. Bridges are formed between two cells. So via these bridges, the exchange of genetic material is happening. So conjugation bridges are formed between the two cells, and via these bridges, exchange of genetic material occurs. And this E. coli, they divide by conjugation. Okay, so this genetic material is passed from one cell to another by this bridges, and E. coli, they divide by the means of conjugation. Now we will move on to the last slide. That is, what are the main conditions which are necessary for bacterial growth? So we should know that, right? So temperature should be, the ideal temperature for bacterial growth should be 24 to 45 degrees centigrade. While some bacteria, they may grow at a low temperature as 0 degree or as high as 80 degrees centigrade. That is, in hot springs, for instance, they can also live. Okay, so that is higher than 80 degrees centigrade. Now, most of the bacteria, they flourish in air, that is aerobic kind of bacteria, and bacteria, they can also survive in absence of air, that is anaerobic bacteria. Now when it comes to the pH level, bacteria, they mostly flourish at an alkaline, slightly alkaline pH condition, that is pH should be 7.4, and however a few bacteria, they can tolerate extreme acidity as well as extreme alkalinity. Okay, so ideally the pH should be 7.4, but they can tolerate extreme acidity or alkalinity. Okay, so this is all we should know about bacteria, and we have already spoken about reproduction, their shape, their structure, so their mode of nutrition and all. So now in the next slide or tomorrow's class, we will be discussing about the economic importance of bacteria and what are the harmful effects of bacteria. So for knowing what are the economic importance of bacteria and what are the harmful effects of bacteria, you should subscribe to my channel to get the daily updates, regular updates. Please stay tuned, stay updated with my channel, and please do share it with your friends so that they can also get the updates which I am posting. Thank you.